Hello, good morning, girls. Now I'll start with the immune response, the next chapter after the complement system. Okay, so you've already learned about B cell receptors and T cell receptors. So anyway, I will just discuss about that because it is given separate in the chapter. So what is B cell receptor? BCR, which consists of an antigen binding transmembrane immunoglobulin. BCR, that means B cell receptor, which consists of an antigen binding transmembrane immunoglobulin. That is known as MIG. MIG is a membrane-bound immunoglobulin. So this is in complex with two transmembrane polypeptides. It is in complex with, so this is the membrane-bound MIG. MIG. So this is in complex with the two transmembrane polypeptides, namely alpha, Ig alpha and Ig beta. So these two containing immunoreceptor tyrosine activation motifs. So these are known as tyrosine activation motifs. They are known as ITAMS. That means immunoreceptor tyrosine activation motifs. ITAMS. So which enables the transmission of the intracellular signaling. So these two. So these are the ITAMS, which enables once the antigen binds to the receptor, which enables the signaling. So this is the structure of B cell receptor. BCR consists of an antigen binding transmembrane immunoglobulin. So this is in complex with the two transmembrane polypeptides, namely Ig alpha and Ig beta, and containing immunoreceptor tyrosine activation motifs. This helps in signaling. So you can see here the structure. This is the MIG structure, membrane bound immunoglobulin. So along with that, the Ig alpha and Ig beta, Ig alpha and Ig beta, and these two are joined by disulfide linkage, and these helps in the signaling. So this is the general structure of the B cell receptor. B cell receptor. So next one, T cell receptor. So the T cell receptor, B cell receptor and T cell receptor, there are so many differences between B cell and T cell receptors. So the T cell receptor, or it is also known as TCR, is a molecule which is found on the surface of the T lymphocytes. So B cell receptors are found in the B lymphocytes and T cell receptors are found in the T lymphocytes. So this is responsible for recognizing the antigens which are bound to the major histocompatibility complex, that is MHC molecules. So the T cell receptors are meant only for recognizing the antigen which are bound in the major histocompatibility complex. So this is the structure of T cell receptor. Which is an heterodimer which consists of alpha chain and the beta chain, so linked together by a disulfide bond. This is the disulfide bond, and which also consists of a constant region. This is the constant region, so it is similar to that in the immunoglobulins. So constant region, this constant region anchor the receptor to the plasma membrane. This is the plasma membrane, okay, and it also consists of a variable region. This is the variable region which contacts with the antigen. So this is an heterodimer which consists of alpha chain and the beta chain, which is linked together by a disulfide bridge, and the constant region anchors the receptor to the plasma membrane, and the variable region which anchors the antigen. So this is the structure of the T cell receptor. So each T cell bears TCRs of only one specificity. So one T cell which is specific for one antigen. So this is known as allelic exclusion. So each T cell bears the TCRs of only one specificity for only one antigen, which is known as allelic exclusion. So this is the structure of the T cell receptors. Both alpha and beta chains have a variable region and the constant region. So V regions of the alpha and beta chains contains the hypervariable regions that determine the specificity for the antigen. So alpha and the beta chains have the variable region and the variable region and the constant region. So V regions of the alpha and beta chains contains the hypervariable regions that determines the specificity for the antigen. So the role of the MHC molecules. What is the molecules bind and transport the antigen fragment to the cell surface? Cell surface, a process which is called as antigen presentation, can then detect the antigen fragment displayed on the cell surface. So uninfected cells, MHC molecules bind and transport the antigen fragment to the cell surface. This process is known as antigen presentation. A nearby T cells can then detect the antigen fragment, and depending on one